All right, hello everyone. This is Michael, and my bacteria, my microbe that I had for the semester was Staphylococcus aureus. All right, here are my models that I presented in class last week. We'll talk about those more later, but let's look at the PowerPoint. So Staphylococcus aureus is a prokaryote cell. It is part of the bacteria kingdom. It is multicellular. It has a cocci, which is a spherical arrangement. Excuse me. Staphylococcus, which means it has a grape-like cluster arrangement, as I kind of depicted here with my models. And again, you can see here with the image of looking through it through the microscope. They can range from diameter anywhere between to about 600 nanometers in diameter. Cell wall, cell structure, and cell protection. Staphylococcus aureus is a gram-positive cell, a microbe, I'm sorry. So when you stain it, it is going to stain purple because it is gram-positive, okay? The cell wall itself is anywhere between 20 and 40 nanometers thick. It has a peptoglycan that serves as a protective barrier within the cell wall. And it also has polysaccharide capsule that surrounds the cell membrane, a cell wall. And within the cell wall, there are protein A and adhesions, okay? And let's look at my model here. As we can see here, the outer portion is going to be that polysaccharide capsule. On the inside here, we have the cell wall, okay? Intercellular components, all right? It contains a double-stranded DNA, as depicted here in the PowerPoint, okay? It also has MECA, which is a gene that makes the microbe resistant to antibiotics such as methicillin, penicillin, and other penicillin-like antibiotics, all right? They also have plasmids inside of them that carry resistant genes and viral-resistant genes that can pass through the population, or like say, from bacteria to bacteria through a binary fusion. I hope I'm not jumping ahead, through horizontal transfer, okay? And they also have ribosomes that perform protein synthesis, and they translate genetic code, and they're gonna have other enzymes and toxins, all right? So let's look at my model. Here, we're gonna have my double-stranded DNA. We have a plasmid, and we have some ribosomes, all right? And again, that is my cell wall along the outside, surrounded by polysaccharide capsule. Cellular metabolism. Staphylococcus aureus is a organotrophic heterotrophic, so it means that it does not make its own food. It has to get its food from another organic source. It uses glucose for cell growth, uses potassium in metabolic processes, and the metabolic processes that Staphylococcus aureus can perform are aerobic, anaerobic, and mixed fermentation. But it prefers, excuse me, aerobic, which is it needs oxygen, or prefers the presence of oxygen. We have coenzymes and cofactors that are going to be used for Staphylococcus aureus. Coenzyme A, that's going to replace glutathione, I believe I pronounced it right, as primary low molecular weight phenol. Cofactors that produce exotoxins that activate host zymogenes and exoenzymes. Cellular reproduction. They reproduce by binary fusion, okay? The size of one circular chromosome is roughly 2.7 to 2.8 mbps, and the average length of the genome is roughly 2.7 million BP. All right, it has 28 genomes. It can replicate roughly every 1 to 3 hours. That's said to be in the nose. It uses conjugation to facilitate DNA transfer into bacteria, and it can form transduction and transformation. It is a mesophyll, so it means it best grows in temperatures ranging from 15 to 45 degrees Celsius, and it is also facilitative when we talk about the pH level that it prefers to live in. It can live in a very acidic environment, so it shows here 4, okay, and it can also live in a very basic environment, shows here 10. And the optimal range is roughly 6 to 7, which is about 15% of sodium concentration. Staphylococcus aureus strands and pathogenesis. It has roughly 110 strands. It is an opportunistic pathogen. Most common staph infection is the ball. Okay, can cause the following diseases and illnesses: impetigo, cellulitis, food poisoning, bacterium, toxic shock syndrome, septic arthritis, and pneumonia. It is part of our normal flora, so it can be found on the skin and in the nose. It can also be found in the normal flora of livestock. It is pathogenic when the microbe can enter inside of the body through a break in the skin via a surgical incision or through the hair follicle. It is a communicable pathogen, which it can be transmitted through skin-to-skin contact. Microbes that are contaminated come in contact with a mucous membrane. We have contact with body fluids, inhalation of contaminated droplets of the saliva, and contact with a contaminated vector. It could be bread sheets, bandages, things of that nature. It is the most common HAI infection or illness that is out. It can form biofilms on inanimate objects such as surgical tools, venous catheters, urinary catheters, contact lens, heart valves, pacemakers, and prosthetic joints. You can also, like I said, get it from contamination of clothing, bed sheets, bandages, or PPE. So it is important that we dispose of those things. How to prevent the spread of Staphylococcus aureus. The autoclave is the best way to sterilize equipment. We can also use decontamination disinfectant. They are both effective strategies that can reduce Staphylococcus aureus on the surfaces of some cases, and boiling can also be effective, but the most effective way is going to be the autoclave. We can also use aseptic techniques and universal precautions as both practices to reduce the risk of the spread of Staphylococcus aureus. And there is a antibiotic resistant strand of Staphylococcus aureus, most commonly known as MRSA, which is methicillin resistant Staphylococcus aureus. It is the cause of a staph infection that is difficult to treat because of its resistance to some antibiotics. There is no current vaccine for Staphylococcus aureus. However, antibiotics that are commonly prescribed to treat staph infections include certain cephalosporins, such as cefazolone, nefelicin, oxacillin, vancomycin, and daptomycin. And we also have taflovacin or linazoid. And those are the antibiotics that can that work well against staph infection. And that's pretty much it.